Hello, it's me. I was wondering if after all these years you'd like to meet to go over.
Spermatogenesis is the formation of sperm cells. The process takes place in males in the lining of the seminiferous tubules. Spermatogenesis begins during puberty and continues throughout life.
The production of sperm cells begins with a diploid germ cell called a spermatogonia. Spermatogonia are stem cells or undifferentiated cells that divide meiotically. One daughter cell remains a stem cell, providing a renewable source of sperm, while the other specializes into sperm.
During G1 of interphase, the spermatogonium does its job. It synthesizes proteins, lipids, and carbohydrates needed for the cell to grow and function. During S phase, the cell replicates its DNA. During G2, additional proteins are synthesized. The cells also begins making organelles and molecules needed for cell division. By now, the spermatogonium has specialized into a primary spermatocyte and is ready to undergo meiosis I.
Meiosis I begins with prophase I. During prophase I, the chromatin in the nucleus condenses to form chromosomes. Each chromosome consists of two sister chromatids connected by a centromere. Next, the nucleolus and the nuclear membrane disintegrate. The centrioles move towards opposite sides of the cell and the spindle fibers form and attach to chromosomes. Homologous chromosomes line up next to each other in a process called synapsis. The homologs then cross over and exchange parts. The sister chromatids are no longer identical.
After prophase I is metaphase I. During metaphase I, homologous chromosomes line up along the equator of the cell. The chromosomes align randomly so that paternal and maternally derived chromosomes may end up on either side of the cell. This process is called independent assortment and helps ensure genetic diversity.
The next phase of meiosis I is anaphase I. During anaphase I, homologous chromosomes are pulled apart by the spindle fibers and moved toward opposite poles of the cell.
Once the chromosomes are separated, telophase I begins. The spindle fibers disappear and the nucleolus and nuclear membrane reappear. The cell is divided into two genetically different daughter cells and the chromosomes become chromatin.
The primary spermatocyte has now divided to form two haploid secondary spermatocytes. The secondary spermatocytes undergoes interkinesis. During interkinesis, these cells manufacture additional proteins and DNA is in the form of chromatin. However, DNA is not replicated.
After interkinesis, the secondary spermatocyte begins prophase II of meiosis II. During prophase II, chromatin condenses into chromosomes. The nuclear membrane and the nucleolus disintegrate. Centrioles move to the poles. Spindle fibers form and attach to the chromosomes.
Next comes metaphase II. During metaphase II, chromosomes line up along the equator of the cell.
After metaphase II is anaphase II. During anaphase II, the centromere split and the spindle fibers pull the sister chromatids apart, moving them towards opposite poles of the cell.
Once the sister chromatids are separated, telophase II begins. The spindle fibers disappear. The nucleolus and the nuclear membrane reappear. The chromosomes become chromatin, and the cell divides into two haploid daughter cells called spermatid.
Each spermatid develops a flagellum. The base, or mid-piece of the tail, contains mitochondria to provide the sperm with the energy for movement.
The spermatid also develop a membrane-covered head called an ecosome. that produces enzymes to help the sperm penetrate an oocyte and fertilize it.
So hello from the other side. I must have called 1,000 times to tell you.
The end. Bye bye.